For my final project in my microbiology lab, I was given an unknown sample of bacteria and had to use differential testing to determine the identity of the bacteria. Hi, if you're new to this channel, I'm Sydney Brooks, a senior at Arizona State University studying speech and hearing sciences, and this is Science with Sid. The objective of this experiment was to isolate a bacteria from a sample broth and use the isolated colonies in further testing that reveals its biochemical properties. These results will be used in a dichotomous key, a flowchart that uses the results of several tests through the process of elimination to determine the unknown bacteria. Without this vital information on the bacteria's properties, in the medical world, incorrect treatment methods or chemotherapy may exacerbate the harmful effects of a pathogen's effect on the human body instead of eliminating it from the patient. This experiment will be conducted over several days to give the bacteria enough time to incubate and grow in its new environments after inoculation. On day 1, I conducted an isolation streak using aseptic technique, one of which I have done many times in the past in our previous projects. I used an agar plate, inoculation loop, Bunsen burner, and a marker to label my sample. Using a sterilized inoculation loop, I streaked the bacteria through 4 different quadrants on my agar plate and set it to incubate at about 25 Celsius for 24 hours. On day 2, we constructed our dichotomous key that would be used to guide the testing of our experiment. We were given a list of 8 possible bacteria that were the identity of our unknown experiment, and using the information in Burgie's manual, I was able to create a flowchart to guide my experiment. I was originally planning on doing a gram staining on this day, but because the results can be obtained in a single lab session and I still had multi-day tests to perform, I decided to save this test for a later day. Taking my incubated quadrant streak, I now had the isolated colonies to use in further testing for my experiment. I decided to continue down the left side of the flowchart, assuming that my bacteria was gram-positive, so I inoculated a methyl red Voges Prosker, or MRVP, test tube. This tube will be used for 2 different tests on day 3. The next test was the sulfur indole motility test, or SIM, where I aseptically used an inoculation needle of bacteria and stabbed it cleanly through the center, making sure it wasn't all the way to the end. Using a citrate medium, I swabbed a needle full of a colony and slid it along the slant, trying not to puncture it. The next 3 experiments were similar in needing 3 pre-completed test tubes full of a carbohydrate, inverted Durham tube, and phenol red as an indicator. This test assesses the bacteria's ability to ferment and metabolize the carbohydrates inside of it. I aseptically inoculated some bacteria into the tubes for lactose, arabinose, and mannitol. All of these tests were set to incubate at room temperature for a couple days. Day 3 of the experiment was mainly gram staining, evaluation, and other confirmatory testing. I took the MRVP tube that was inoculated, vortexed it, and used a 1 milliliter pipette to transfer it into an Eppendorf tube labeled VP. This is where we separate the tube into the 2 different tests, methyl red and Voges Prosker. I added Barrett's reagent A and B5 to the tube, vortexed it, and allowed it to sit for 30 minutes to see if results were shown. I also added some drops to the MR side of the tube for extra confirmation, but the results were not necessary for determining the bacteria's identity according to the dichotomous key created. While the MR and VP tests were cooking, I conducted a gram stain using the sample, loop, Bunsen burner, microscope, stain tray, babyless paper, DI water, and stain kit. The stain kit contained crystal violet, Graham's iodine, decolorizer, and safranin as a stain for gram-negative bacteria. This test shows us the properties of the bacteria's cell wall and identifies it as either gram-positive or gram-negative. Starting from the top, the bacteria looked like this under the microscope, pink and rod-shaped. This classifies it as a gram-negative bacteria and has a thinner layer of peptidoglycan surrounding each of its cells. This layer is important in its susceptibility to antibiotics. The VP testing showed no color change, indicating that this bacteria does not produce butanidol as an end product. For the SIM test, the black color present shows that it produces sulfur from the media. A positive indole test would be a red ring at the top, but being as there was none, it does not produce indole through the fermentation of tryptophan. For the citrate test, the dark blue color change shows that it can use citrate as an energy and carbon source. This is important because it helped us differentiate from Citrobacter frundi and E. coli as seen in our dichotomous key. Now at this point, I was fairly confident that the bacteria was Citrobacter frundi, but I still needed to analyze the final confirmatory test to accurately determine the bacteria's identity. These three tests were important in showing which carbohydrates it was capable of fermenting and using as an energy source. For the PR lactose test, it changed color from red to yellow, meaning that it can ferment lactose as an energy source. The results were consistent with the tests for mannitol and arabidose. The MR testing shows its ability to produce lactic acid from glucose fermentation. While this test was positive, the results were only used to rule out any other possible bacteria. So, what did we find? Using our key, the unknown bacteria was Citrobacter frundi, a group 5 gram-negative bacteria in the rod shape. It is usually found in soil, water, and the intestinal tract. In humans, CF can be found in the urinary tract because it can be dense in citrate, a primary energy and carbon source for the bacteria. Its properties as a facultative anaerobe give it the qualifications to survive in areas of the body that have little to no oxygen and can be classified as an opportunistic pathogen, meaning that it can infect a healthy individual if they become immunocompromised. In the real world, CF is being utilized for bioaugmentation as a soil stabilizer due to its ability to anaerobically fix nitrogen and phosphorus into ammonium and phosphate. This is currently being used at airfield and airbase operations to reduce the amount of solid organic waste in the soil, as well as promoting a cleaner way of waste removal and soil recycling. Because CF is gram-negative, antibiotics are usually very effective against it, but overuse and overstimulation may be causing this bacteria to have gained a resistance to antibiotics in some places. I hope you learned a lot about this testing and about Citrobacter frundi. Thank you for watching Science with Sid. If you enjoyed this video, check out these next videos because YouTube thinks that you will like them.